Okay. So with our esteemed panel up here who have really shared a lot of knowledge this morning, we've covered quite a breadth of material. A little insight from myself, I'm a brand owner. You know, everything that was discussed, you know, is part of our job to make sure that we figured this all out. With a lot of these parameters considered, these different perspectives, these different approaches, it's all pieces of the same puzzle for me at the end of the day. So I found it very interesting. Really, all the talks contributed a very different perspective that all really get to the same thing. And as I was sitting here reflecting on it this morning, I thought, you know, I was pretty comfortable with packaging when I came in today. I'm not so sure now. There's always more and more things to think about. So before I ask any questions or we come up with some of the other questions, what I would like to do first is open up the floor for any questions that you had relative to any of the specific talks that you didn't get a chance to ask, or maybe that's something you thought of on break or while having a discussion with somebody else. So does anybody have any questions for any of the panelists really specific to any of their talks before we maybe open it up to the broader audience or panel?
Now look at your notes.
Here we go.
So Paul, this is a question for you. I did learn something, even though I've heard that presentation many times. It's always interesting, you pick up new tidbits, but with your definition of color additives, and then you said that those color additives can be used in food packaging pretty much at will if it's approved as a direct food, directly added to food as a color additive. Does that, could that be said as well for all food additives can be used? So it's just a specific for color additives can be used in food packaging directly.
Yeah, thank you for that question, John. No, that is specific to color additives. A direct food additive cannot be used as a indirect food additive without an approval for use as an indirect food additive. But there is a distinction for that as well for generally recognized as safe substances. For the majority of substances that are generally recognized as safe for use as direct food additives are also considered generally recognized as safe as indirect additives as well. So if it has a listing in 184, for example, by 184 I mean Title 21 of the Code of Federal Regulations, Part 184, it can be used as an indirect food additive as well.
I might add, relative to the color additives, you know, I talked about using some sophisticated instruments. When it comes to color additive, we don't need any of that. We just use our eyes. If that color additive turns that food color, it's not allowed. And recently, that just happened. In the lab, someone was doing a test, and they were mixing a sample that was a food oil and a colored plastic, and they were looking for something else. And I said, well, why you keep mixing this thing? And they said, well, I haven't finished the time yet. I said, as soon as it changes color, it's done. It's not allowed. So you don't need an instrument for that. You just your own pair of eyes.
Thank you all for your talks this morning. I really enjoyed all of them. Jim left us with his last slide he had, can safety be conveyed in a manner that transcends geopolitical boundaries? And I like that because of the, as he mentioned, incongruence that leads to compliance gaps. In terms of toxicology, you know, the UN came out with GHS, globally harmonized system. And most countries now have begun to approach it and incorporate it in some sort of way. Do you think the terms of safety, though, from the different approaches that Jim mentioned, dealing with migration or dealing with exposure, dealing with toxicology, do you think that there is an approach that we could harmonize, such as even if we, with the vehicle, let's say, for example, ISO?
Let's take that one first.
Okay.
I think it's a little, not unfair, but it's a little difficult for a regulatory body of one place to talk about, like, well, how do I make it work for other places? So, but I would like to set the stage with that. We struggle with this all the time in regulatory companies, and because the packaging that's approved in one place is not only being used in that one geographical area, it's being packaged and used all over the world. So we do need to have these commonalities, and I think it's similar to what Tim talked about. It's hard to prove a negative. It's almost like, well, okay, we know there's some differences, but let's find where there's some common ground, and then be able to say, this is where it translates. Like the model for food contact it's, was it 1 kilogram is 6 decimeters squared, and for FDA it's 1 gram is 1 square inch. There's a mixture of 10 grams in 1 square inch, 10 grams in 1 square inch. So it comes out to, if you break it down, it's about the same, right? The one comes out to be like 10 grams and 6.5 centimeters or 6.0 centimeters squared. So there's commonalities. I would like to hear more about us making those commonalities. Like where do we find those areas within the concentric, the circle, and the square? And then there's little areas that are outside of those bounds, but there's a lot that's inside the bounds.
I think maybe I can follow up from the toxicology side. And I agree. I think there already is a lot of harmonization. A lot of types of tests we recommend follow often OECD guidelines, so it's similar to ISO, and so that is harmonized across the world. And we also participate in a lot of international meetings with JECFA, WHO assessments. We collaborate with EFSA, other groups. So when you see some of the more popular chemicals or more public ones, that's where you have the more overt harmonization, where we're obviously working together. But behind the scenes, we also work together too with collaborations and discussions. So we are working on that. And we try, one of the importance with the new science is to get the new science validated at an international level as well. So it is standard across the world.
I definitely think it's something we need to start moving towards, and yeah, having those discussions around where are the commonalities and are we essentially doing the same thing and are we arriving at the same conclusions. Things like ISO is definitely a good way. However, how that's interpreted in regulation can differ. So ISO standards can appear in regulation, and how they appear can cause divergences. Equally, the political landscape can have a massive influence. So even if the regulatory system is in one way and is harmonized, the political landscape can overturn that. So Europe is a fantastic example of where you can have some very good regulation. However, the decision making can be very much removed. So you've got like the European Food Safety Authority who do risk assessment, you have the European Commission who do risk management and make the ultimate decision. And they can be, BPA is a perfect example again of how this didn't necessarily result in a decision that I feel was a science-based one. So I think that's actually the challenge you're going to face even afterwards. So being mindful of that eventuality and that just reality of it is really important as well.
Just in case it's easier to pass this on.
Thank you all. Appreciate it.
If I can comment on that a little bit. Well, I think, certainly toxicologists done a great job, follow OECD guidelines, and, and the data, I think, largely can be shared whether you submit to FDA or submit to EFSA or any regulatory agency. But I think what's difficult is once we translate into a regulation, that becomes language that can no longer be translated back and forth. So, as an engineer by training, I think the way to overcome reference frame differences is try to find a way to non-dimensionalize it. Just take it in a way that, irrespective of any kind of system that you use. What do I mean by that? If we if we go from toxicology and then start talking about safety factors, we can say safety factors of 5,000. I think that's a universal language, whether it's in US or EU, right? Instead of saying this is compliant. I think if both places will not only talk about the regulation, but also give as much publicity, if you will, to the safety factors that are already in there, then that becomes a translatable language. That becomes translatable from one to the other. I know we do that, but but people just don't seem to appreciate the power of that kind of simple metrics. I mean the general public. I think I'll just stop there.
Any other? I know my life would be exponentially easier if the world could agree on one system. What I get faced with a lot is, well, if it's safe here, why is it not safe there? And my answer is typically, I'm not saying that it's not safe there. What I then have to say is it's not regulatorily compliant there, which doesn't necessarily, to go to Jim's example of the SPET, for a while, I mean, ultimately it's safe, right? The FDA said it's safe, so it's safe, but you still couldn't use it in Europe because they hadn't come to their own same conclusion with a different set of data for a different set of rules. So it would be much easier.
And a question about the exposure yardstick that's being used to measure the migration or transfer of components from different types of packaging into food around the globe. And I'll say one thing is, to piggyback on that, is I think we need to think about the future and want to get your thoughts of it for a harmonized system to evaluate food packaging in terms of the amount of materials that can transfer to the packaged food, and also to have more predictive tests that represent reality. I think one thing I saw is Dr. Grob's work up on the screen, and he's actually published some articles that say certain types of food simulants and those sorts of tests grossly overestimate the amount that goes into food, which is great if you believe in the precautionary principle. But if you believe in the realistic principle, and what Congress said is, hey, we need to have a reasonable approach. How do we get more reasonable testing methods that really predict how much go into food, and what could be done to encourage that both in the States and around the globe?
I'm the only chemist in here. That's a good question. And I think there is opportunities to work on some of that harmonization from the migration and exposure assessment side of things. And I know we've actually had some discussions with our Health Canada partners, as well as at least Luigi Rossi was involved with the EU, that we had some discussions with him as well. And you can imagine, the fits and starts you have with talking between countries. But I think there is some room for that, and actually I was really interested in your presentation about the tools that you've developed, and I think there's some collaborative work that we could actually do to give some of our data and actually start looking at how we might be able to use that particular tool in our evaluations. So I think there's plenty of room.
I mean, it's interesting as well because exposure, as being, if you think of it as being part of risk assessment, it's the dynamic and changing part of it. Okay, the hazard is an intrinsic property. The substance exposure is a real-world phenomenon, and that, by its very nature, varies from geography to geography. People consume different foods; they use different packaging. So you kind of need to allow for that equally well in your risk assessment. So I think And I'm glad you raised the precautionary principle, because that's going a level deeper and thinking about the value system that underpins a regulatory framework, which if that's not harmonized, then regulation is always going to diverge as well. So asking those very fundamental questions when you're trying to look at harmonization, I think is really important.
And also specifically to the United States, yes, we do have guidance documents which list generally applicable types of either migration solvents or exposure constants to be used to calculate exposure, but those are generally applicable. If you have information on the specific use that you have for your product and you want to limit that product to that specific use, you can come into FDA pre-notification consultation, explain the reasoning, the scientific reasoning behind either you want to do a different simulant or you want to use different constants in your exposure calculation, and as long as it's scientifically supported, we can evaluate it and provide you an opinion back of yes, you can either use that for example, your future FCN, or no, we don't agree. Just because it's in the guidance document doesn't mean that's what you have to do. That is just an option. If you have scientifically supported information for an alternative approach, we're more than happy to review that information, and then if it is supported, it can be used to support a notification, for example. So if you have those type of questions, please come in for a pre-notification consultation. Thanks.
So this is kind of a general question. So when we talk about substance exposure from the FDA and maybe from the European perspective, do we consider like combinations of additives? I mean, a class of substance, for example, of phthalates. So we know there's several phthalates, right? Taken together, maybe you get one effect as opposed to individuals. You get an additive effect. So considering something like that, and then we're also seeing phthalates contaminate actually our food supply. So now that's another exposure route. It's actually in the food. It's not just coming from the packaging. Not to say we're using phthalates, but that's just an example, in general.
That's a multifaceted question.
It's the reality we're looking at.
It is the reality. I mean, you've woven in a toxicology question in with an exposure side of things. So as an exposure chemist, I can look at, and we typically look at compounds, individually, A, B, and C. You calculate an exposure, and then the toxicologist can work their magic on those numbers. So from a standpoint of an exposure side, I could do a cumulative exposure to various materials, provided that our toxicologists need that type of an assessment. So we can do those types of things from, a mathematical standpoint, but it really drives down to what's going on the tox side. Do you want to lump compound A and compound B together? Is there some physiological, biological reason to do that?
I'm so glad you mentioned biological, because my question to probably the FDA in general is, this whole area of bioactive substances, that's kind of keeping us awake at night, at least from the consumer products goods company side of things. How are we looking at, this is like a new world, right? It's the whole BPA question. It's not toxic, it's not a toxic substance, but it's biologically active, has potential endocrinic effects. So where are we? Help.
From that side, we can separate that into mechanism versus toxicity. And anything that you eat is going to affect some type of mechanism. If you eat tofu, it's going to affect your estrogenic system, right, or endocrine system. So it's really getting to, often our recommendations and the type of studies are very broad, capture mainly toxicology, because even if you do have a mechanism activated, you have other mechanisms within your system that can counter that, can balance that, the whole idea of homeostasis. It's not until you push those mechanisms beyond a certain level that you start to build up toxicity, and that toxicity builds up and builds up. And that's what we try to capture in the broad tox testing studies. However, we are looking at mechanisms now. There's a lot more studies becoming available. A lot of the in vitro studies, cellular studies, all look at mechanism. And those provide supporting information to give, like I was talking about before, a more, I say our assessments are accurate, but more precision within that assessment, a better understanding. And maybe moving forward, like I was saying before about in my talk about taking all the in vivo data we have on the tox side and all this new data and starting to make those correlations to where we can see what point or what mechanism or multiple combinations of mechanisms could be predictive of eventual toxicity. But that's something we still need to work on. We still are working on.
If I can just go back a little bit on the comments about exposure, I think it's a lot of undertaking for CREM and under the auspices of the European Consortium and the Commission and for the FDA to do. But I think the industry itself can do a lot of the work, printing ink and laminating adhesive, for example. The industry probably have very much the same toolbox and pretty much know the exact quantity of chemical that are actually used and probably can come up with a very good exposure model and actually come to a very precise exposure assessment. And that in turn will give a lot of credence as we to the regulatory agencies, whether here or Europe or anywhere, how far away we are from the ADI, how far, how many order of magnitude we are actually today, so that we do not get unprepared to respond to the questions of safety factors. So something come up, we can say, we actually have the realistic model that actually can answer those questions. Today, I think we just call it NIAS, and then we start doing the worst thing we can do, which is basically do the hazard analysis and then just throw up our hands. I think it's not the agency's role alone. I think it's the industry that need to coalesce and do its job.
Just two quick perspectives. On the question of multiple sources of exposure to a single chemical, in Europe, EFSA are calling for all sources of exposure to be considered. So the same chemical can be present in food contact materials. It may even exist as an additive or a flavoring. Equally, it can be present in personal care products, cosmetics, other consumer products. And they want to see that factored into an exposure assessment. Equally, the Scientific Committee for Consumer Safety, who regulate cosmetics, want to see dietary sources of exposure to a chemical considered, especially for CMR substances. So that's something that I've been busy with a lot lately. On the question of multiple exposures to multiple chemicals, the cumulative question, I think there's a very good framework in the US at the moment for pesticides. I think that's a good guiding light in terms of how they manage multiple potencies and multiple sources of exposure and relative potencies. In Europe, I think they've cocked this up completely for pesticides. EFSA have a common action group that can have up to like 200 chemicals in it. So when you're trying to do a risk assessment, you're stuck straight away. So again, I think the bioactive question is precisely the thing you need to ask when addressing that, even when you're doing your exposure assessment.
Since many of you are from the FDA, so I just take this opportunity to ask the question about labeling. As a requirement, each ingredient or additives directly added to the food must be listed in label, right? And however, if we add ingredients or additives into packaging material, and the packaging material we release the additives or ingredients to the foods. So the label must be listed these ingredients or additive, and if so, how much? Because, for example, if we add 1,000 ppm something into the packaging material, and the packaging will release, but may not only list 10% or 50%, or even not 100%. So the question is about label. We need to list the additive into packaging. If so, how much we need to be listed in the label.
So the answer to that question isn't dependent upon the amount that goes into food. The question is that component being released from the packaging in order to have an intended technical effect on the food at the point of consumption? If it is, then it has to be included on the label. It doesn't matter what level it is. If it's not, then it's not a food additive. It is a food additive, but it's not a food ingredient and would not be included on the label. So,
however, if we say that as an ingredient, for example, salt, we not treat the salt as a food additive. We treat the ingredients. So how about we insert the salt into packaging material, let the salt release to the food? This is treat the additive.
If you add salt to food, you have to include it on the label. So it doesn't matter if you're just sprinkling it on the food and then packaging the food, or if you had it in the package and it transferred from the package to the food. It's intended to have an effect on the food, therefore it has to be labeled.
How about amount? See, to my understand that the label must have the largest amount listed first and go through to the trace, right? But for example, we added 1,000 ppm, maybe directly into the juice, for example, sodium benzoate, and the list the third one or third one, the third one of the ingredients. However, we added 1,000 ppm sodium benzoate in the packaging material. Packaging material released may be much lower than 1,000 ppm. So they should be moved to the bottom of the ingredients list, something.
So there's sort of maybe two parts to your initial question, and maybe I didn't realize that. So there's no threshold of where you would not label something if it is added to the food in order to have a effect on the food. You would have to label it no matter what. But you are correct that things added to the food are listed in concentration order, and that would be taken into account as to where that particular additive would fall on the list of ingredients in the food.
Okay, thank you.
You wouldn't label what was intended to affect the orange juice in this case. I think that's yeah.
From a packaging perspective, you'd only label it if it has an intended technical effect on the food.
Saying, okay, if it comes back and preserve the orange juice, then we have to label it.
And Dennis, you look like you have something to add.
Yeah, I think that question was only relative if you're talking about an active ingredient. So you have an antioxidant in the packaging film, and it's actually formulated. So they're using tocopherols, for example. So it's formulated in the package in order for it to become an antioxidant. And so I would think, wouldn't they have to do some type of migration studies to understand over the shelf life and use of the package, how much migrates out, and then that would tell you where it would be labeled in the ingredients. I think that's the only example of what you spoke about that's applicable here, it's an active ingredient made to diffuse through the packaging film, become a food additive to protect it, to extend the life cycle, for example, the shelf life.
Okay,
So I think that's exactly right. In that case, if you were actually doing that in order to have an effect on the food, you would have to do studies and you would have to anyway, to determine how much is going into the food, and then that would base where it would fall in the ingredients list. But as Dennis said, none of the FDA people up here are labeling experts. That's actually a completely different office than the one that we're in. Right. So, if you have specific questions, contact that office.
Well, if I can, I think the what you're asking is already in place today. I think Greg mentioned, BHT added to cereal liners, and BHT, that use is indicated on the cereal liner box. So I think, if you say, how about things that are below that? Well, then question is how, if you want to have every single component that's ever used on packaging to be listed, then, you're probably no container will ever hold enough information. And then you actually introduce more exposure from the printing inks to the food. So, that's the reality, right? And that's, again, bear in mind that packaging itself, the exposure is orders of magnitude below that of edible food. It's probably on the orders of the flavor ingredients, or even fractions of that. And we don't list every single possible flavor ingredients in the list of ingredients. We just say natural flavors. I think that's perfectly okay under the labeling regulations. So, it's simply not practical, or for that matter, useful to ensure consumer safety.
Okay, since a lot of our talks this morning really focused on chemistry and toxicology and some exciting new developments and analytical methodologies, I think the first question up on the slide is something that almost every panelist could address in a few sentences. Kind of maybe we could just work our way down the line. And because I think the interesting part is, as our chemistry, which alluded to in one presentation, gets better and better and better, what do we do with this information? I found very interesting this slide about how you had that chromatogram that was just a mess, and then it interpreted what those things were. Well, if you let the public know that you found all those things, then again, how do we assess and utilize this information for real safety assessments of our packaging materials? And I mean, we have toxicology perspective, we have chemistry perspectives, we have analytical methodology. If maybe we could just kind of go down the line and speak a few sentences of what you see, how we can use this. We're better in the lab, but just because it's there doesn't make it bad. Something along those lines. So if Greg, maybe you could start.
Or maybe Tim would start.
Okay.
You don't mind me volunteering to put that?
Well, it is true that our analytical capabilities have gone so far, and we can now measure so little, that I think it's, relative to tox perspective, that's for sure. We actually just yesterday had a question from a different section of FDA that's actually interested in this. And they asked, well, what examples do we have about where this happens? And one that I know most people in the room here probably are somewhat familiar about, we had the Deepwater Horizon oil spill. And that contaminated huge sections of our seafood harvesting area. Same thing happened with the Exxon Valdez. And then it is FDA's responsibility to make sure all that seafood is safe. And when you look at the concentrations, and in order for us to open up the seafood fishing in those areas, we look at the main metric was for pHs. And we can measure pHs in incredibly small amounts. But the tox evaluation and their health hazard evaluation is thousands of times above what we can measure. That's actually true. And so now, we're in this situation. I know Kirk and other people there have to deal with some of these low numbers, but we can actually measure incredibly low numbers. Now, as far as the nanomaterials, there is some interest there. How do we evaluate the safety of those? We've actually done a lot of testing on some of these materials, and are they released from packaging? And there's a size effect there that we know about. And basically, a lot of those materials are relatively large compared to regular molecules that are released, and they tend to be released not at all. I mean, it's very difficult to measure nanomaterials that actually come from packaging material. they've put nano silver in packaging, and initially what happens is you can measure a little bit of silver in the food, but then it stops, and that's basically because it's nothing more than a surface effect, and that material can't move its way through a packaging material because it's too big. And so as far as other packaging materials, how much is enough? That's just we're going to have to deal with that public image of that we can measure very small amounts, and the exposure is a lot less than we can actually measure in the lab, for sure.
Please, yeah.
I think that's an important part about the public perspective, the public message, what's being interpreted in the public. And I can give a few examples here. I think it's fantastic that the analytical chemistry is getting so good, the limit detection is getting so low. But the idea is that, oh, okay, we found all these things. They must all be bad and terrible. And no, those limit levels are much lower than would be expected to cause any toxicology. But the public perception, I'm sorry, the public perception is there. It's more of a perception of hazard. And I give you an example. So with studies with, didn't want to mention this word though, but BPA and BPS, we've had some studies where we looked at both of those, and they've been presented publicly as, okay, in this material we found, or in the blood we found BPA and we found BPS, so it must be terrible. However, there was a very big difference between limited detection looking for those two separate chemicals. It's much, you can get much lower limit detection looking for BPS because there's a lot less contamination background from BPS than as you would from BPA, so limited detection is higher. And so when I've seen some presentations of this, they're saying, oh, look, we found BPS too, it's just as bad as BPA. But when you look at the exposures, and they usually don't like to show this, is that the estimated exposure from BPA was here, while BPS was so low. So when you're talking about bioactivity and potency and comparison, action on mechanisms, you wouldn't expect them to have the same action because they aren't at the same levels, although it's coming across publicly that as long as it's there, it's a problem. And from the other side, I want to say about a good benefit of these increases in analytical methods. We had examples where things have been on the market for years. They've proved decades ago, and the methods were a little bit different then. And you get an exposure assessment, sometimes based on limited detection, because the product or chemical might not really even be there, but there's a limited detection. That stops us being able to go completely to zero. Recently, we've had some new submissions come in where they wanted to use this material. It's been used for a long time, but redid some of the analytical methods and used a much lower limit detection. And from what our previous exposure estimate was, we've been able to reduce that. So now we get at a more realistic level, and it's not as exaggerated. We're talking about getting more realistic exposures, using better methods to get more precise measurements. And that's what we're seeing in some of the resubmissions from products that were regulated and analyzed years and years ago.
From the standpoint of the supply chain that's part of making the packaging article, I find it, and an analytical chemist, I find it to be getting to the point where it's going to be very, very difficult to claim that something is free of. So phthalate-free, BPA-free. The lower and lower the detection limits go, we can go down to parts per trillion, femtograms. And what we're going to do is now that it gets detected, then no one will be able to claim that something is free of or that that chemical is not there, and I think it'll drive a frenzy that should hopefully collapse itself because it can't be sustained that you can continue to look at everything at a very, very low level when it really is insignificant in terms of the risk to the consumer, the risk to the public, even the risk to the environment. And I think that that's the challenge, is to be able, for the technical community and then the regulatory bodies also, to be able to almost, you want to intercept that public perception, that public rollout, so that that little quip that they state is already addressed, because we see it first. We see the technical information first, but it's really a big challenge. I really see it as that. I agree. One of the things with looking at that particular question, it always hit me back to the Paracelsus, the dose makes the poison. So it's not just chemistry, we can go way down with our analytical processes, but from a tox side, where do you want to stop? And the concept of, threshold of toxicological concern, we're working on refining that process with work in our office, can we refine it more? Are there particular things that, it's maybe an aflatoxin-like material, you could have a threshold, okay, if they're aflatoxin-like materials, we really need to go down to this level. If it's something like diethyl ether, that's going to have a whole different type of a threshold. So being able to think about this and perhaps like that TTC type concept will help us to say, okay, we can stop now.
That's a great idea, I think, to be able to group these categories from the toxicological concern standpoint. That's excellent, because that would help a lot with the industry itself along the supply chain.
Yeah, just to say, I totally agree. I was going to suggest the same thing, in fact. I swear. But taking a pragmatic view like that, and so I totally agree that the consumer misperception is driven by hazard, and just because they think it's there, they think all hell is about to break loose, or that their food supply system has been compromised. And at the risk of sounding like a broken record, it is the exposure that contextualizes whether that detection is actually relevant and has a human health risk or not. In terms of practical tools, using things like screening level type calculations and threshold toxicological concern can give you a way of just either of quickly screening whether these measurements matter or not, and whether you need to do anything further with them. So I think that's a quick win that we could get with a modest amount of work.
I just I think we're all thinking alike. Well, I was thinking alike as well. TTC help contextualize the forests or peaks and make it relevant. But I think, we also touch on, as we pursue analytical limits, and then, where does toxicology, what kind of limit are we pursuing? With that said, I think what's missing today is some kind of concept that's, we learn in physical chemistry, which is the uncertainty principle, right? There's time and space, and there's some definite, there's a quanta below which there's nothing. You cannot get it you can only be certain up to a certain limit. And I think what today we don't have is the equivalent of the Heisenberg uncertainty principle for chemistry and toxicology. These two are together, but we are pursuing one without recognizing that you're losing on the other side.
Question?
Yeah, actually, just a comment. I just wanted to follow up on the TTC approach. I think that's our best hope actually moving forward, and because the chemical space is just so huge, right? So you're going to be detecting all sorts of new molecules, and as to how do you justify, how do you defend safety? And I think that the TTC approach is the most promising tool that we can use, and I hope that we can all come together and try to push that forward. As you all know, the TTC approach actually branched out of the TOR approach. So I just think that's something that we should all support, for the benefit of of the food industry in particular.
I had a follow-up question. This question, I really like this, and I like all of your responses so far. If we kind of put that together, just yesterday, Erin Brockovich happened to be on NPR, and she was talking, and she went, "Oh, such and such is in the water at 1,000. Oh, I forget the units." And then she goes on and she said something about, and this is present at 1 parts per trillion. And it was like, Okay, so with
Huge number.
huge amount. So with this in mind and lessons learned from BPA, it's been published now that we have real risk. That's a factor, of course, between hazard and exposure. But there's perceived risk, it's which we need to deal with, which is the public's perception of the risk, which is the hazard, followed by outrage. And we can only control the outrage. And this is going to get to be a bigger and bigger problem because zero keeps getting smaller. If you were to pick up a newspaper today, nobody would believe it that the year I was born, the water and the air was so much dirtier. There was no EPA. And now it's cleaner than ever. But if you were to pick up a newspaper, and as most people do, it's driven by media that's just hazard. So as industry and as government and as scientists, how do you propose that we control not the hazard, but the outrage?
Well, there you go.
Yeah, let me answer something else. You hit on the major points there. It's the public perception. It's what we were talking about recently, and it's one.
You should just move that microphone closer. There you go.
I'm too quiet, I know.
Yeah.
And the public perception, and this threw me off there for a minute. The goal, one of the big challenges, we're talking about challenge toxicology, challenges in chemistry, but you hit the major challenge for us all to do, to work together, is public education. I think that's the only answer. It's making sure we're conveying the science in a way that people can understand, not too technically, that only we can understand certain cases, but it's the public education, the public outreach, putting the hazard into perspective of the exposure and explaining exactly what that means. I think that's all we can do. You can't control people, can't control outrage, but you can try to stay calm and explain the science as best as possible.
We did propose at dinner last night that we should start blogging more, as others seem to do, and seem to help control that outrage or throw gasoline on it, whichever is your perspective.
I think that's a really good idea, actually. Genuinely, if you want to make any ground in the public sphere, you got to engage in the actual battleground where these arguments happen, and a lot of scientists are utterly removed from where the misperception of risk actually happens. So I would say people actually do need to get stuck in there on social media. There's a lot of very good science-based blogs that I would follow that are called The Skeptic's Guide to the Universe, IFLS, which I want to explain stands for. These kind of blogs are pushing the public perception and understanding of science, and I think that is the counterbalance that needs to happen, and it has to happen. It's not going to happen in the peer-reviewed literature. Nobody's going to read that, so you're going to have to engage in the actual battleground where this happens, and I think blogging is actually a very good start.
And you need a clever name.
Absolutely. Oh yeah.
Don't go with what you're working with now.
Yeah. And get rid of the PhD at the end of your name.
So actually, one of the things that I flagged this morning was the talk about the different exposure limits for proposed limits for infants versus adults. How are we going to manage that? I mean, just an open question, because it's not okay for infants, but it's okay for adults. But as a packaging user, all my packaging, I don't know, I don't package just for infants or just for adults. I buy my packaging from a company that supplies me with packaging. So does the infant level become the new de facto level for our packaging?
If I were evaluating it, I would say yes.
Yeah.
Because you can't make that statement to say, well, this would be okay if you were an adult.
Once again, it's that compliance gap.
Right.
Now becomes a exposure hazard gap.
Actually, if I can comment on that, and not, not to comment on presentations that I did not give, but in reality, there is no, the testing tiers are the same. It doesn't matter if it's adult exposure or infant exposure, and that's one of the things I think Jason, or maybe it was Kirk, I'm not sure, was trying to make clear in their slides. It's all a matter of how you express the units. The concentration actually is the same, or I should say the exposure is the same when you're expressing it on a body weight basis. So an infant has a smaller body weight than an adult. So their overall systemic exposure for a given concentration in food is going to be dependent on how big they are. It's like, if you like to drink beer and you're 300 pounds, you can drink a lot more beer than somebody who weighs 150 pounds. There is no difference. It's just how you express the units.
Or what was the question, was that the answer to the question, or is that more of along, you're going to get your material from a manufacturer, how do you know that was approved for an infant style use or another product? And that's part of the food contact notification process is now on, I mean, maybe you can talk about that, Paul, about the additional language on the notification for when we've evaluated for infant food safety.
And from that standpoint, it's also no different than a substance that's approved for use in fatty foods or aqueous foods, but isn't approved for use in fatty foods. Infant foods is just another food type. It's really no difference between, it's just a different limitation on the use. It's the same thing, as I said, for something that's allowed for use in aqueous, but isn't allowed for use in fatty. So you would have to do the same diligence with your materials supplier as a packaging manufacturer as you have to do for any other additive that has a food type limitation.
With respect to the infant, regulation, I guess, that you guys are proposing, I haven't had any real conversations about that, but something that we might consider, which I haven't talked to Kirk and company about. What we did learn from the BPA story was that, initially, I actually did the testing for us. We measured bisphenol A coming out of a polycarbonate bottle according to our regulations. It was there, no question about it. But when we eventually went to reading the instructions on the infant formula container and then mixing it in a polycarbonate bottle and serving it within the time frame of which an infant would consume it, you can't measure bisphenol A. So we may just think of that concept that we may look at it, as a use condition instead of always looking at the worst case. That's something that, but the damage was done after we made our measurement in a polycarbonate bottle many, many years before the whole thing blew up. It's just there. You can see it. They said they quoted the numbers. But when you actually mix up some infant formula in a polycarbonate bottle, shake it up, and feed it to an infant, that's how I fed my kids. It was unmeasurable, and even with low detection limits. I mean, we could seriously measure pretty low detection limits, still couldn't measure it. So it's just a thought that as we formulate the new infant thing, it's something to think about.
No, I think that goes back to what's been said before, is trying to get those more refined exposure estimates. And, as Paul pointed out, our recommendations are recommendations. If there's a different way, a better way, then, we'll take a look at it and come and talk to us about it. So there may be alternative ways of calculating any exposure. So we definitely want to, doing it with respect to our recommendations, you're looking at kind of a worst case. So it's from a safety perspective, that's a good thing. It's conservative, but sometimes you do need to refine your exposure estimates.
So I grew up in the exposure risk assessment world in the 80s. That's where I cut my teeth with doing it. And one thing I've learned that we might think about how we apply it going forward is sort of having sort of screening assessments, then refined assessments, et cetera. So you start out with the worst case, and this is how I think of in doing product stewardship, you do the worst case sort of scenario, and you say, if the worst case doesn't give me an answer that keeps me up at night, that there's harm that's going to occur, et cetera, then I stop there. But if it does, then what I start doing is sort of a sensitivity analysis and say which factors are the most important factors to get a more realistic estimate of the true exposure and true risk and the true outcome, and maybe even the true hazard, because everybody says hazard is inherent to the product, but actually all hazard probably isn't created equal and diagnosed equal, because you can have some hazards that are believed to occur with some materials, but in some species that hazard isn't relevant to the species because it doesn't produce the same effect. And I think we saw some species things, and some people say, well, that's toxicology and that's hazard. But when you also think about the factor of what keeps people up at night, and I'm going to come back to outrage in 1 second, and it's sort of the voluntary versus involuntary. So somewhere in this whole process, we need to go ahead and say, let's refine it around, and you have all the science, and you package all the science, and you say, we continue to bring this in to be the most realistic estimate and the most realistic scenario we have. Then we need to step back from it and say, okay, then what drives outrage if it's involuntary decision, right? We know that. If it's being done to me, I'm going to be outraged. And that's where EPA learned about radon when they started to do the radon in your home. If you owned the home and radon was in your home, people wouldn't test. They had to get people outraged. And so what they did is they said, oh, we'll do it at the real estate transaction stage, right? And so then they said, now I can blame the prior owner so that they've got to pay to fix the problem. I think we need to think through how the public thinks about hazard and risk and how we go ahead and build credibility, because it all comes down to the credibility of these sophisticated mathematical models that you hide behind a black box, right? In Facet, yeah, that data's in a black box. We can't see the factors of the confidential information there. What does that lead to in terms of the public trust? And that's where we probably need to think about is how do we regain the public trust, because it's so bad in some situations that it's political science, not real science, that drives the decisions, like with BPA probably, right?
Thanks. Thank you. With that, I would like to call this morning's session to a close. I'd like to thank all of our speakers for sharing all of their knowledge and experience in these areas, as well as participating in this panel. And I hope that everyone found this of great benefit this morning. Thank you.